Hi, my name is Fenil, and I'm a biology undergrad at the Maharaja Sayajirao University of Baroda in India. And what I'm going to present today is work that I did with my colleague Devin at Dr. Pushpendra Singh's lab at the same university in the summer of 2019. And what we've done is we've made a program that can tell the user what strain of the leprosy bacillus they're dealing with based on just a few inputs of SNPs. So I'll begin by explaining what SNPs are. SNPs are single nucleotide polymorphisms, which are differences of one nucleotide in what are otherwise similar genetic sequences. SNPs can be very informative. They can be used for genotyping of a pathogen into lineages, which can give us insights into their phylogeny and phylogeography. Drug resistance mutations are also often a result of SNPs. Mycobacterium leprae and Mycobacterium tuberculosis are examples of pathogens where SNPs can give us such info. Mycobacterium leprae or the leprosy bacillus is the causative agent of leprosy, and it is an uncultivable pathogen, meaning it can't be grown in a lab. Being an intracellular parasite, it has undergone reductive evolution, in the process of which it has lost a lot of its functional genes and has thus accumulated over 1300 pseudogenes. There's very little genetic diversity that exists between the different strains of this pathogen. And comparative genomics has revealed a few SNPs which can allow us to classify this pathogen into the different strains. And currently there are 4 major SNP types, within which there are a total of 16 subtypes. And these genotypes usually correlate with the geographical origins of this pathogen. So this is the genotyping scheme for Mycobacterium leprae. After you've sequenced a given sample, you can use SNPs at these informative genomic loci to tell you what strain it belongs to. For example, if the SNPs at these 3 genomic loci are G, A, and C, then your sample belongs to SNP type 1. And then, if the SNPs at these 4 genomic loci are C, G, A, and G, then your sample is of the subtype 1C. Identification of SNPs using this scheme manually is error prone and can be cumbersome. What is desired is automation of this type of classification, which can be both more accurate and easier. So this is the program that we have made to automate this genotyping process. It is called SNPSoL, or SNP-based Strain-typing of Leprosy. It can do conventional genotyping, where it identifies strains based on the typing scheme shown earlier, or it can directly check for predominant genotypes that you might be suspecting. These could be geographically prominent strains or newly identified strains such as these. So this is the program that we have written in Java, and this application is BlueJ, which is an IDE for Java. And I'll run the program. So when you run the program, it asks if you want to check for predominant genotypes or go for conventional genotypes. And let's say I have sequenced a sample of Mycobacterium leprae and I have no guesses as to what strain it might belong to. And in that case, I'll go for conventional genotyping. And let's say the subtype or the strain that I have uh, that I've sequenced is 3J. And in that case, I've opened the genotyping scheme here. And in that case, this SNP 1642875 will be T. And so I enter that. And the second SNP 2935685 will be C. And the third 14676 will be C as well. And then it tells you that the SNP type is 3, and then it directly goes into the subtyping, and here it asks for 1295192, and let's say since it's 3J, it's G, in which case it'll ask for the second SNP as well, which is 2312059, and that is C. And that is enough for this algorithm to tell you that this SNP subtype is 3J and the strain that I have with me is 3J. And let's say now I suspect a particular strain or suspect a newly identified strain that is not part of this genotyping scheme. In that case, I'll go for predominant genotypes. And these are the predominant genotypes this program can identify. And against each predominant genotype are shown the SNPs and or the indels that this program will require for classification into that strain. And this is the SNP in the reference genome, and this is the SNP in this particular strain. And so let's say I suspect the strain 4N, in which case I'll choose 4N and I'll enter. Given that it is 4N, 110180 will be G, and 264623 would be A, and then it tells me that the subtype is 4N. So we've seen that the Java program makes the process of genotyping much easier and less error prone, but it still requires you to install Java and a Java IDE such as BlueJ. So what we've been working on currently is we're making a web app using the open source app framework Streamlit using Python. And this can take both manual inputs as well as an Excel file containing all SNPs that you found using sequencing relative to the reference genome, and it can directly give you an output. So this makes it much more user friendly. It doesn't require you to install any softwares. It's online, and if you have an Excel file that contains all the SNPs, you do not need to enter any data. This can be used to identify drug resistance mutations, which can be very useful in clinical diagnosis. And finally, this can be extended for strain typing and identification of drug resistance mutations in other pathogens such as Mycobacterium tuberculosis as well. Thank you for your attention.
Hello. Thank you so much for your talk, Dr. Fenil. So I'll see if there is any question in the chat. Yeah, the video is pixelated. So Sharanya, can you please optimize this setting to 1619? While waiting for the setting, I have a question for Dr. Fenil. Hang on, is Ananda Rizkia is here? Oh, okay, that's not question. Okay, so Dr. Fenil.
I'm not a doctor, I'm just an undergrad.
Okay, Fenil.
Yeah.
It's very interesting. So I want to ask you whether this will be only specific for Mycobacterium leprae, or will you also use for other species such as Mycobacterium tuberculosis? And comparing to other pathogen and also Mycobacterium tuberculosis, how is the genomic diversity of M. leprae, and is it very diverse? I mean, you know that this, as far as I know, I mean, so what drives you to do this? What motivates you?
Oh, okay. So right now the program is for Mycobacterium leprae. And so the lab that I interned in works with mycobacteria and primarily Mycobacterium leprae. But yes, the plan is that it can also be applied to other pathogens as well, which require SNPs for their genotyping. And as for the diversity of Mycobacterium leprae, there isn't a lot of diversity, and you can use SNPs and VNTRs to genotype them. There's no morphological diversity or biochemical diversity in terms of gene products. And so what we use are SNPs and VNTRs to genotype them.
Okay, okay. There's a question from audience. Is there any way to validate the result?
Yes, so the genotyping scheme that we've used is from publication from 2011, which I can send to you on the Discord channel or something. So it's
Yeah, you can
um,
send in the
yeah,
chat as well. It's fine. Or you can also send in Discord.
yeah, Discord, yeah, I can send
Mhm.
it there.
Okay. So will this be available on GitHub or other platforms such as Galaxy?
Probably not Galaxy, but yes, so right now we're working on a web app that we can make available on GitHub later. We just started working on it. Yeah.
That's wonderful. So, okay, an audience asked for file in the chat. So if there is no more questions, maybe we could go to the next talk. Is there any more question from the audience? All right, thank you very much for your talk.